All right, hey guys, so here's this second podcast on photosynthesis for chapter 10. Sorry, it's a little bit late tonight, but you guys were telling me you were mainly watching these during advisement anyway, so that should be okay. Releasing it a little bit late to you guys tonight. Just make sure that you check it out before you come to class tomorrow. And this is a fairly short section, 10.3. Not a lot of reading, but it's really, really dense with a lot of material. So I wanted to make sure to cover this pretty thoroughly for you guys. But I think probably the most important thing to start with is what's important. Why are we learning so much about photosynthesis, and why is the College Board focusing so heavily on this, and why is this even something that you need to learn? Because you might be sitting down thinking, I don't really care that much about plants. This has no relevance to me becoming a doctor. Why should I care anything about this? And really the main reason is when you guys go to college and you start taking your big fundamental biology and chemistry courses, one of the big focuses is on where does carbon come from, right? So where does this atom specifically come from as you're tracing around the ecological level? Where does that ultimately come from? That's the big question. So to get to the relevance of why we're learning this stuff, I'm just going to start with a few examples. This right here is just an image of the molecule serotonin, which is responsible for several things in neurology and in your nervous system, but most importantly, probably right now, it's 9:32. You should all be going to bed right now. If you're getting tired, this hormone serotonin gets released into your blood system. And notice that it has 10 carbons. So it's C10H12N2O. That's the formula of serotonin. That carbon needs to come somewhere. It needs to come from somewhere in order for you to make serotonin, in order for you to feel sleepy, in order for you to regulate your circadian rhythm, in order for you to wake up in the morning and kind of get refreshed. So that carbon ultimately needs to come from somewhere. And again, the question in AP Bio is, well, where does that come from? Where's the ultimate source? Right. So here's another example just at a level above the molecule level. We're looking here at neurotransmitters and receptors, and this would be how serotonin is sent throughout your nervous system in these small little vesicles in which that molecule would be trapped. It would receive a receptor. The backbone of all those molecules that make up all these neurotransmitters and these transporters and the axons and the terminals and the synaptic vesicle and all of this stuff ultimately has a backbone on carbon. So there's more carbon that we actually need. Another thing that we just learned about in the last unit was just the phospholipid bilayer, in which we see all of these fatty acid tails, the hydrophilic and the hydrophobic regions. All of that contains carbon. Right? So the ultimate source of where all this stuff comes from is from CO2. So that's why there's this main focus from the College Board on why you need to know that, and why we focus on photosynthesis. And it also will help us when we get to the next unit, which is ecology. So last podcast, what I focused on was the light reactions. And now the second part, what we're focusing on is the Calvin cycle. So a different cycle than the citric acid cycle or the Krebs cycle that I covered with you guys in oxidative phosphorylation and cellular respiration. This is just the second stage in photosynthesis. So make sure to separate those out. And then what I've got written out in here is just the balanced equation for photosynthesis, in which I've highlighted in green here the two key components of what's going on in the light independent reactions or the Calvin cycle that we're focusing on. So yesterday you guys saw that these 6 water molecules are going to lose their electrons, right? They're going to get oxidized and go over to oxygen. So ultimately this carbon dioxide is what we're looking at today, and this glucose and how those relate to one another. Really, so CO2 is going to get fixed into glucose, and we're looking at how that process happens. So this is just a diagram that I had in the last podcast also that shows you the system here, the ecosystem level, which energy is flowing in photosynthesis in the form of light energy, and that's involved in the light reactions. And then CO2 and H2O are going to be your inputs into that, and out comes organic molecules. I'll explain why they say organic molecules and not glucose specifically here in a second. And then oxygen gas, and those are going to be then the inputs to cellular respiration, where when that energy gets processed, it's mainly lost as heat, so it's transferred. Here's the location that I wanted to point out of where all this is happening. These right here are the thylakoids that are stacked into stacks called grana, which is something that you should know, but mainly the thing that I'd want you to know is that these are the things that are containing chlorophyll, and that's why they have that green color, and they contain the chloroplasts. All of this space outside of these thylakoids is where the Calvin cycle takes place, which is what this image is trying to show you. That CO2 is an input. This is the location of the Calvin cycle, and then the output is G3P or the sugar. This was the diagram that we used to describe the light reactions yesterday, in which the main things that we had were step one here, you had a splitting of water to drop off the electrons to photosystem 2 that get excited by those photons. The electron transport chain in these photosystems, they're going to move along, and that change in delta G, the movement, the free energy that's now available from those electrons being passed along, going from high potential to low, drives these H pluses, these protons, against their concentration gradient and starts to stack up in the thylakoid space. So you have a huge concentration of H plus here with only one escape to go through this enzyme ATP synthase that phosphorylates ADP and inorganic phosphate into ATP. Then those are going to be one of your inputs there into the Calvin cycle. So that energy that ultimately came from light is transferred here into ATP, and that's going to go into the Calvin cycle. So that's one part of where the energy is going. But then also those electrons need to be dropped off. So where they're going to go is they're going to get excited again here in photosystem 1, and they're going to be passed along in order to take NADP plus and reduce that to NADPH. So this electron shuttle is now going to be an input into the Calvin cycle as well. And then your waste product here is a little bit of oxygen gas. Right, so we've traced those, and now we're looking at, well, specifically, how do you take an inorganic form of carbon, which is in CO2, and move that into an organic form? And that's this absolutely terrifying looking diagram that you have sitting in front of you right now. The good news is this looks fairly complicated, but you guys really only need to know really 3 basic phases. We can chunk it up into phases for what you really need to know. And what I did is I just wrote a few notes down of the main components that you guys should know in order to break down this image. So I'm going to break this down step by step. Phase 1, carbon fixation. Phase 2, reduction. And then phase 3, regeneration. And then I'll come back to this image, and hopefully that will be a little less intimidating once I break this up into phases. So in this first phase, this yellow phase that we're looking at right here, there's a few important characteristics and reactions that you guys do need to know. This one's just called carbon fixation. So this is the movement of CO2 from the atmosphere into something that's actually a usable form. So what happens here is a molecule of CO2 combines with this 5-carbon acceptor molecule called ribulose 1,5-biphosphate, or what we can just call it is RuBP. That's what we're going to just trim this down to, so we don't have to say that complicated word every single time. So this CO2 is going to combine with a 5-carbon acceptor called RuBP. So it's like a little shuttle that's going to grab onto the carbon dioxide and brings it along. RuBP is the bus and CO2 is the passenger. The step that makes a 6-carbon compound that splits 2 molecules of this 3-carbon compound called 3-PGA is here in this step. So this 6-carbon RuBP and CO2 complex, there's now 6 carbons, gets split in half to this thing called 3-PGA. That reaction is catalyzed by this enzyme called RuBP carboxylase. You don't need to know that either. We'll just call it rubisco. So that's what this little enzyme is right here. That's the enzyme that they're showing you. So this is the fixation of CO2 going into that RuBP, and now you've got a different product, right? So then we get into this second phase. So what really happened in phase 1 with all that complex language? CO2 got incorporated into a substrate. That's all that happened. In step 2, you have reduction. So what we were looking for was that CO2 would eventually get reduced into glucose, and that's where this step happens. This is ultimately where the electrons from the light reactions are going to go. They're going to reduce CO2 into some usable form, some organic form of carbon. So this is the second stage, in which ATP and NADPH are going to be invested, and they're going to take that 3-PGA molecule that we made in that first stage, and they're going to move those into something called G3P. Now, G3P is sort of the main output of this system. So glyceraldehyde 3-phosphate is really just a precursor that could make any sort of sugar, right? So this G3P, if you stack 2 of those together, what you can get is a 6-carbon sugar, or something like glucose. Put 2 G3Ps together and you have the basic ingredients in order to make sugars. Right? So this stage really gets its name because that NADPH donates electrons or reduces the intermediate, the 3-PGA, in order to make the G3P. So again, like all that language, what are we really doing? We're taking some of that energy that we made in the light reactions, we're taking the electrons, and we're just going to transfer those to G3P, or excuse me, PGA, in order to make G3P, which is the precursor for all the sugars. So now that we've got our product actually made, this is a cycle. So we have to go back through and we have to make those substrates again, and this is probably the most complex portion, phase 3 is. In really simple language, what we're doing is we're taking some of that G3P and we're going to make some glucose out of that. And then we need to regenerate the cycle, which requires some ATP, which is what this part of the image is showing you here. So through a series of very complex network of reactions that luckily you don't need to know, we're going from G3P, some leftover G3P, up, back, and around to this ribulose 1,3 biphosphate. So all of these little arrows are showing you separate reactions that would eventually lead you back up to that intermediate, back up to that substrate. The big take home then from that entire cycle is that in order for one G3P to exit the cycle, which can go on and become glucose later on in the plant, 3 CO2 molecules are going to need to enter the cycle, and that provides those 3 new atoms of fixed carbon, right? So those are the things that are actually going to move into the sugar. When 3 CO2 molecules enter the cycle, what you get ultimately is 6 G3P molecules that are made. And I'll show you this next image which helps with that a lot. One is going to exit out of those 5. One exits the cycle and is used to make glucose, while the other 5 you have to recycle those to regenerate 3 molecules of the RuBP acceptor, which is where we started. So right here is a more friendly version of what I was just describing to you guys, in which we have the input here of 3 carbon dioxide molecules, in which rubisco is going to help bring those inorganic forms of carbon into this intermediate. And now we have 6 carbons sitting right here. There's your intermediate with 6 carbons. This gets split now into 3 carbon molecules. This is your 3-PGA, in which we have 2 molecules of 3-PGA coming from this one 6 carbon intermediate. Right, so we have a total of 6 3-PGAs from these 3 CO2s that were originally there. We're then going to invest that ATP that we got from the light reactions and the NADPH. We're going to oxidize that down into NADP plus in order to reduce our substrate here. Now when we do that, when we go from the PGA and we invest all that energy in those electrons, we're going to turn this into G3P. So we get one G3P that actually goes on to make glucose, one 3 carbon molecule that goes on to make glucose if you stack 2 of them together. 5 then of those 6, remember we had the 6 up here, 5 of those are going to be moved, 5 of those G3P are going to be used in the regeneration phase, in which we'll invest a little bit more ATP in order to get 3 RuBP molecules, which are each 5 carbons long. So that's the entire cycle that you're going to need to know here. My expectation then tomorrow is that there's a lot of questions on this. This is a super short video and a very quick run through in order to understand a really complex topic. There's a lot of biochemical pathways here and a lot of biochemical reactions. So tomorrow, if you guys have questions or if there's anything that I can explain, please bring those to class. Also, one of the great things about the videos and doing these lectures on video is I'm about to be done with this in about 13.5 minutes. In class, this would take probably 45 minutes. So we can spend those 30 minutes just answering your questions that we normally would just hear me boring on and on and on about a specific topic. So hopefully you guys are watching this the next day. You're watching this on Wednesday. None of you are still up. But if you are, make sure that you're writing some questions down and you're bringing those to class with you tomorrow. Okay, so I hope that's helpful, and I will see you guys tomorrow.